initially as a monitoring editor and now as an associate editor. So I have a chance to see an awful lot of different manuscripts coming across my plate. I've been on editorial boards of quite a few other journals as well. So that you get different kinds of manuscripts and see different aspects. And here at the Danforth Plant Science Center, I'm also on the Committee for the Responsible Conduct of Research, where we run an RCR program for the entire center.
Oh, I see. All right, thank you very much for your introduction. And as Toby said, there's a lot of aspects about research integrity and research conduct and misconduct. And today's our topic, we focus on the data over-interpretation. So let's jump right into the first question that I would like to ask our two panelists. Now, in your experience, when and why this data over-interpretation happened? Maybe starting with you, Toby.
Yeah, I think it's a good question. I like the way the question is phrased, because over-interpreting data is a completely natural human thing. I mean, you develop a hypothesis and you get very attached to it. You're excited about, you think, well, okay, cool hypothesis. Let me do an experiment and to see if that hypothesis is true. And because you're excited about the hypothesis, you sometimes forget about either designing the experiment in a way that will tell you if the hypothesis is wrong, or you just, find things that will support your initial hypothesis and kind of forget that maybe there are other interpretations of the data. So I don't think it, by itself, data over-interpretation is so natural a process of the human brain that, we see the human brain is very good at detecting patterns to the point that it will detect patterns when there aren't any. I mean, for instance, in Northern European cultures, people will look at a full moon and say, oh yeah, it looks like a face. Well, in other Asian countries, people will look at the full moon and they'll say it looks like a rabbit. So, it's not that, and of course, neither one is correct.
Right.
There is no man in the moon or a rabbit in the moon, I don't think. It's just the way our brain operates. And so the challenge, I think, in science is to build in as many ways as we can, as many layers of checking as we can, to consider the possibility that you're wrong.
Yes.
Basically. And that part of that goes back to designing a really good experiment. The really good experiments are the ones that are going to tell you something, even no matter how they come out. And even that is a challenging, designing an experiment like that is challenging. But yeah, at every step of the way, and particularly if you're working on something where you're very fond of a hypothesis, it's sort of,
all of us are doing that.
Yeah, it happens. And so somehow training yourself and then putting yourself in an environment where somebody's going to say, hey, Kellogg, you missed this. That hypothesis really might be wrong. And so I think it really starts right at the beginning of the process.
I see. Thank you for that. Larry, do you have anything to add to that?
Yeah, I think that definitely there's a human element to it there, and the bias that we all carry with us. And on top of that, I think there's also a lot of institutional burden on researchers to basically sell a story or to create hype around the work and excitement. Sometimes people talk about it in terms of impact, and because we all need this for our career and promotion, there's also some external forces that may push us to over-interpret what's actually there. And I think that when we say over-interpretation, we're saying that we're kind of making a bigger deal out of our results than what is actually there. maybe you're extrapolating outside of the context of the research or the plant species. Like it's common to extrapolate from Arabidopsis, say, to all crop species or all plants.
Yeah.
And at the same time, extrapolating from one environment to another, like say from a greenhouse to the field. So there's many ways to over-interpret data due to both our own bias, but also we're being in some way pushed to do that. So it's not only up to us to put the brakes on, but I think it's also up to our supervisors and managers to, when they evaluate us, to evaluate us more fairly.
Mm-hmm. And I think it probably pretty obvious that the peer review process becomes one of the last checks on this. Is that a peer reviewer, if they're really paying attention, which they often do, peer reviewers are generally going through and saying, well, do I believe this? Has the author made a convincing case for whatever they're presenting? And so I think peer review is kind of close to the final step and before a paper gets published.
Mm.
But it should be just one of many points of checking yourself,
Yeah.
checking your data.
Yeah.
Agree.
Right. So yeah, talking about seeing faces and seeing rabbits. When I see that in my data, I mean, SDS page gel chromatograph, it's time to leave the lab. But all jokes aside,
Yeah.
After all, are we allowed to freely wander in our own realm of speculation from the data? How harmful is it? I mean, should the audience be able to judge for themselves where the results end and where discussion begins? Can I just freely speculate and the readers would have to see through that because they are all scientific readers and they're all scientists? Should that be the case, or over-interpretation is really harmful and we have to avoid it at all cost?
I think there's room for speculation, but it turns up in sentences like, from these results we might speculate, and actually use the word speculate.
Mm-hmm.
Obviously the most famous one of those things is, it has not escaped our notice that this structure, can't remember the whole quote, gives an obvious mechanism for its replication, which was not part of the data in the Watson-Crick paper, but that was
for the DNA structure.
Yeah, exactly. That was the paper that described the structure of DNA, which was a very, very short paper. And the very last sentence is saying, well, this does show an obvious mechanism for how it might be copied in the organism. But they didn't produce any data on that, nor did they, they didn't even claim that they had. I mean, there was nothing over-interpreted there. It was just, a speculate, it looked kind of obvious that might be the thing that was going to happen. So I think there is room for speculation, but it has to be quite clear that it's taking a jump beyond the data.
Mm-hmm. Yeah, I think in all of our journals that the results and discussion sections are separate in the journal articles, which helps. And I personally feel that type of discussion and speculation does belong in the discussion and not in the results. It's harder in some journals where the results and discussion are mixed.
Yep.
I personally don't like that style, but I know that a lot of people do.
Mm-hmm.
Yeah, so I just saw one question from the audience, which is I think very relevant to what you have been discussing. The question is, would you say a paper is only ready when you have converged on a single module or explanation, or is it acceptable to present the data and then propose a series or many multiple explanations for that set of data? How would you view that? How would you take those manuscripts when you review them?
I think
starting with you, maybe Larry.
Sure. Yeah, I think different for the different society journals. At Plant Direct, we really focus on the sound science aspect. And one thing that I've seen is that we also accept transfers that were rejected by our partner journals like Plant Physiology or Plant Cell. And these are often very good papers that include reviews. And when they include reviews that are pretty much positive about the soundness of the paper, what I generally see is that the manuscript may need just minor revisions for placing it in the appropriate context. And if there are alternative explanations like the participant is asking, I think it's fine to state those. And the important thing is to state the limits of your experiment. And it's also fine to propose what additional experiments would be needed. So I think that's perfectly appropriate. And I would hate to stifle people's creativity and ability to speculate, because I think that is a big part of science, is running with your results and taking those leaps of where they might go. But just when we put it into the literature, we have a responsibility and a duty to place it in the appropriate context.
Yeah, I would echo what Larry just said, that it's, I hope it's okay to propose alternative models, because I've certainly done it. And I think it's actually helpful to say, here's, assuming that the data themselves are solid, then to say, well, these data are consistent with the following one or two things that could be teased apart by additional information or additional experiments. And I think that's a very reasonable way to, conclude a paper.
Right. Yes. So assuming the data are solid is a whole not a topic.
Yes.
Yes. I hope we have time to touch on later. So talking about when you review those papers, who should be the first person to flag any extra, over-extrapolation or over-interpretation? Because I know that you, Larry, and Toby, you have your own research program, so you're very busy. And there are many manuscripts going through your desk, I assume. And so what are the rule of thumbs that you would go about when trying to flag these issues when reviewing papers?
Well, I would hope that the issues come up in the conversation in the lab before the paper is even written.
Very good.
That somebody has a result and they present it at lab meeting. And I would hope that other members of the lab would, or me, as a PI, would be able to say, hey, wait a minute, you've just taken a step beyond what the data actually show. And then when people write papers, you would hope that they get multiple readers before they're submitted to the journal. So again, there's another set of checks. I think PIs definitely have a responsibility. And I think being busy doesn't let you off the hook. I think it's you're in the, the buck stops here or whatever. If I miss something in my lab, that's my fault.
Yes, that's very true.
Yeah, I agree with that as well. And also during the peer review process, I think it's editors typically look at sort of the big picture of a paper before sending it out for review. They might be able to catch some of these issues, but it's probably more likely that they would be catched by the reviewers, and the editors are definitely relying very, very heavily on reviewers.
Yeah.
I think it's important to say now, though, too, because I can also see that reviewers can be very critical about these points as well, to the point of, getting personal in their statements. So I would like to encourage people as a reviewer, even when you find flaws in a paper, don't make it personal. You can assume it's a honest mistake and try to focus on the science and put it as lightly as possible. Any criticism, there's a person on the other end reading that review. We don't want to turn off our young researchers to science at all because of this, but at the same time, that is the job of the reviewer.
Mm-hmm.
Yeah. So yeah, it's interesting you mentioned young scientists, because there are many people. I remember me when I studied graduate school. It's very difficult for someone who just started to tell the difference between sufficient interpretation and over-interpretation. And so should that be an important component of the training for scientists to realize these issues? So as Toby said, we should stop it in the lab before submissions. So should that be a strong focus of the training programs going forward? And in your experience, how best to do it? Should you involve your graduate student postdoc to in your reviewing process, or you have training seminar, or what works best in your opinion? Starting with you, Toby.
Well, I think that the thing that works most effectively for me is when other members of the lab or other colleagues read papers and comment on the work as it's going along and then as it's getting written up. Because I think it takes more than just one person stepping in and saying, here's a sufficient interpretation, here's too much. I think it's a learned thing, and maybe I'm just slow, but most of us take multiple repetitions of that kind of thing to get a sense for where that boundary is. So I think there's a lot to be said for involving as many people as possible. I certainly know some labs, and my lab is done for a number of papers, often where we just put something up on Google Docs or pass around a Word document and get everybody's feedback on it. And, sometimes it's just people catch typographical errors, which is good because I don't like them. But also people, start thinking about the interpretation, and I hope, if we've built our labs well, there are an awful lot of smart people there. And so I keep telling the people who work with me is that the PIs actually don't know very much. It's about all the brains are in the postdocs and the grad students and the technicians. And those are the people whose input you want in terms of interpretation.
Yeah. How about you, Larry?
Yeah, it's great. Like Toby said, to include people in a PI's group. And another way to do that is getting them involved with peer review for actual papers, which generally, if a PI was requesting themselves to review, they could either suggest instead to invite, especially a postdoc, to review on their own. But I think that most editors would be interested if a PI writes back and asks if they can review with their student and share it with their student, in which case you usually just have to specify in the review that both people led the review and that it wasn't shared outside by those people. But that's also a really good chance for different stage researchers to get involved with the peer review process and to learn about what type of interpretation is reasonable. But again, I also still feel like that a lot of times the most critical people can be earlier career before I think you do get taught in a lot of graduate level classes to be critical of work. I remember some of my classes in ecology where at Penn State, where we read the classic papers in ecology, and basically like we were tasked with trying to find holes or alternative explanations. So I think that type of coursework is valuable as well. So I hope that lots of people get exposure to those type of classes where you read papers and discuss them.
All right, that is a very good idea. Yeah, I think we should start that on our campus. And my background is biochemistry of natural products. So I try to elucidate genes, and sometimes I have to restrain the temptation of claiming this is the gene instead of just saying this is just a gene that could possibly do that. So that I think that is the problem with over extrapolation, and that is just one example. And I think Larry mentioned this before, that you've seen examples of people extrapolate research on model organisms such as Arabidopsis into other plants that have significant industrial or agricultural applications like crop plants. So do you, and Toby, do you see these examples a lot during your editorial experiences?
Mhm.
You did?
Yeah, I mean, I think that comes to sort of the hype aspect that we're asked to state broader impacts, right, of our work. And because of that push, you might, you can go a little too far, basically. So it's just that it's such a fine line. There's and probably for different people, that balance when they are reviewing a work or reviewing its potential for impact. Not only the researcher will have a different line, but the evaluators will too. So it's extremely hard to find that balance. But for me as a researcher, I've always thought of things and only in their context. That it's extremely hard to extrapolate beyond the single experiment that you did.
But once again, I think it's a question partly how you phrase it. That is, if you say, well, I found working in Arabidopsis and I found a gene that's going to enhance yield in soybean. That's definitely a step too far. If you say, well, this gene is causing Arabidopsis to produce more seeds, and it would be interesting in the future to see what its function is in soybean.
Yes, yeah, right.
It's a different phrasing, but it's very clear at that point what you are and aren't claiming about that gene. And I think it particularly is an issue for those of us who do basic research, which is everything that I do is in non-model systems, is that it's not irrelevant to crops, but I'm not working on the crops.
Yes.
And sort of saying, okay, this is the underpinning of a larger picture, can be a little bit challenging. And I totally agree with Larry that we get pushed to say, well, what's the application of what you're doing?
Yeah.
Well, next week there's no application. I would like to hope in 5 years there is an application, but
right. So going back to, yes, that's a very good point. So it's not totally irrelevant. It's actually very relevant if we conduct more research into this, but we're not working directly on those things. However, as Larry said, we are under pressure either from our department head or research program or university, or even from our relative over Christmas dinner. So what do you do? You try to, so, and one question that is relevant to this from the Q&A is that the headline of Institute press releases are regularly over-interpreting your data, and they claim they have wire claims, and how, so the audience, these questions wants to ask Larry and Toby, what's your thought on tamping down those high-profile occurrences that might be less than desirable? So do you have any thought on this?
Well, I don't think you're in control of it. Most of the, when that happens, you talk to, well, you may not even talk to a press officer. They may just look at the abstract of your paper and just do something with it. So, it's certainly, I suspect Larry has too. Many of us have had, and you perhaps have as well, Don, people taking your research and saying, well, Don's going to find the coolest compound that's going to be the next cure for cancer. And obviously you wouldn't mind if that were true, but it is, well, I don't know all the compounds you work on, but I suspect there are very few of them that actually fall into that category. And you know, some of it you just can't do anything about. You can respond to it if you're in a position to respond, but I don't know that it's productive to start writing letters to the editor and telling the press office to back off. I don't know. Larry, do you have thoughts on how to deal with that?
Well, I don't know about dealing with it, but I do think it's an issue that's getting worse.
Yeah.
I've noticed that more recently, that as we move more into social media, you know, I think that, you know, like the clickbait type of headlines
Yeah.
are getting worse. Yeah, and sort of different institutions are in competition with each other, and they also want to highlight their researchers. So I think it's a real issue. It's not one I've seen discussed very, very much. I think it would be worth, if any senior researchers feel like they're in a position to have those discussions, you know, at the higher level with the media outlets at their institutions. I mean, you can't do much about like a newspaper, but we should be able to do something about, you know, our own outlets. I think in my case, I've generally been allowed to look at any type of press release from an institute that I've been at. So I hope that we all have that opportunity. But I know that due to normal journalism practice is often that the people that were interviewed for an article do not get a chance to see it beforehand, because it's not supposed to be influenced in that regard for journalistic integrity. So then that's a whole other barrier, you know, that we may run up against. So I think it's really good to raise this question, and I hope that it's one that people will think more about.
Yeah, I think we need more scientists or researchers to talk about this. I think the issue is quite rampant in animal research, in human physiology. They even have a very popular Twitter account that put "in mice" in every headline. You know, the research claimed this, but you have to remember this research are done in mice, not humans. So I'm not sure we have similar things for plant science, but maybe we should. So, for example, compounded treat cancer, potentially treat cancer.
Right.
That is something that we try to look at. Yeah. So going back to the the over-interpretation. Sometimes I think it's not so much about over-interpretation, but they incorrectly interpret the data, especially when it comes to significant differences. So let's say p-value, for example, some people or many people believe that as long as the p-value is less than a certain threshold, then you can totally claim it, or even it's higher than the usual threshold, they can claim something like my data have the tendency towards significant or something along that line. So in your editor's role, have you seen a lot of these in plant sciences, and how you normally deal with them? Maybe starting with you, Larry.
Yeah, I mean, I think this is something that reviewers are trained pretty well on, because it's such an easy flag to look at the p values. But I know there's also a huge amount of debate about the relevancy of p values and our obsession with it. And then one of the things that got brought up in the lead up to this panel was on the use of false discovery rate, which is sort of a newer idea, extending p-values that's actually more conservative, and that it might be something that we should do more often. It's more common in bioinformatics, because so many statistical tests are done in bioinformatics. You do have to be careful to control the discovery rates. So I feel like this is something that reviewers can catch, but that's certainly more training in statistics would be useful. And now as we see more Bayesian statistics that doesn't have the reliance on p-value, I know that people run into questions about why they don't include the p-value if they use a Bayesian approach. And it's really difficult because most plant biologists aren't a mathematician or a very strict statistician. So the way to get that right can be difficult.
How about you, Toby?
Yeah, I mean, Larry's right. This is something that reviewers can flag and maybe in a better position to flag than, within an individual lab, just because I think many people have had perhaps one course in introductory statistics where what they've learned is you want to do a comparison, you use a t-test and you use a p-value of 0.05. And then that's the end of the course. So that's the tool that they bring to the data analysis. And, that's reasonable, but it's an education process. I mean, certainly I see papers where people use multiple t-tests where they need an analysis of variance, again, to control that significance level. But if you've never been exposed to the analysis of variance and why you do it,
Yes.
I don't know where it was just an education process. And I think it sort of goes back to how people are trained. And in certain fields, some people, like in Larry's field of ecology, it's very heavily statistical. So I suspect anybody in that field has had a lot of training in ecology, whereas historically something like molecular biology was very qualitative. It's changed a lot over the recent decades, but historically it was very yes or no kind of thing.
Yes.
There wasn't the statistical training as part of coming up in that field. So I think the only thing one can do is assume that the author has made a good faith effort to analyze the data the best way they know how, and then the reviewer, the editor, can write back and say, hey, there's a problem with this and here's how you fix it. I think that's a very constructive use of the review process, because that's what reviewers are for, is basically one of the functions is to provide a constructive solution to what they see as a problem.
Yes. Yeah, I've seen some biomedical journals, they even require the authors to send their data set to an independent statistician before submission. Just like you should send microarray data to a database before submit your paper. So do we have anything like that? Do you involve a statistician in your reviewing process if you see a strong or a big component of the paper has to do with statistics?
That's where the selection of the reviewers comes in, that you would try to select a reviewer that has the relevant expertise. And so if it is heavily in bioinformatics or statistics, then you try to find that right person. Yeah. And I'll add that it is becoming, there is a bigger push, to share data and statistical code. It's something that I've been doing since I began my own lab in 2017, that for every paper I share the raw data as well as the R code for it. And I don't know how often people use it. I can see that they download it. I've never been contacted with a correction, but I would welcome, if anybody ever has any improvements to let me know, so I can use that analysis in my future work.
Maybe your instruction is already very clear, so they don't have any questions.
No, I doubt it. I'm sure that there's so many ways to analyze and interpret data that I'm sure that people could find ways to improve on it.
Yeah, so I have one question from the Q&A, and it's related to what we talked about earlier about the pressure of publication. So the question is about publication in crop science, and if your result doesn't show any yield increases, it's very difficult to get published in a high impact journal. But it doesn't mean that the research is not of high quality or is not meaningful. Could you please comment on this? Is yield increase a big thing that the editors look at when you review or you decide to accept or not a paper that discuss or work on crop plants?
I
Toby,
think plant physiology isn't specifically a crop science journal, so we certainly get plenty of papers on crops. I don't remember the question of yield increase coming up anywhere.
Right.
No,
It's
in plant physiology would be, did you find the gene for it? That's
yes,
always what they want to know.
exactly. Yeah, no, there's much more emphasis on finding the mechanistic underpinning of whatever it is that you're studying. And in fact, just because of the focus of the journal, increasing yield is, I won't say it's irrelevant, but it's of secondary importance to understanding what the underlying gene or physiological mechanism
Right.
is.
I think there's been some recent examples of papers in I don't know, I can't remember if it was like Nature or Science, with maybe increasing photosynthesis and leading to increased yield. That I've seen some of this discussion on Twitter, and it may be what they're referring to. Then there was also discussion of how, in actuality, for a yield trial, the data presented wouldn't really be sufficient because it might be like a single site for 2 years and things like that. Whereas to really establish yield benefits, you'd want to see like a multi-year, multi-site evaluation. And then just a larger question of, I think if you do work in crops and agriculture, it can be hard to publish in journals like Nature and Science for whatever reason. And maybe they mean that the only way you could would be that if it's some really outlandish claims about yield increases, but as it turns out, I mean, I think that, for example, Field Crops Research is a journal that I highly respect, and I think that they actually can be very strict about making claims for those type of reasons.
Right. Yes. Right. So yeah, that's very interesting. So if you don't have high yield, then you the high yield you expect, maybe you frame your discussion in a different way and focus more on the mechanism behind it, and we still can publish in high impact journal without having to torture our data to make them confess the thing we want to hear. Right. So
I mean, the data are what they are, and I think, yeah, you don't torture the data. And if they're not, for whatever reason the paper isn't, doesn't have enough replications or whatever for the journal, then you just go back and redo it or change the claims accordingly.
Yes. Right. So that's very interesting, and I think we can move on to other part or the aspect of data over interpretation. So I have one very interesting question from the audience, and the question is about part of the problem with over-interpretation, maybe the way things are written or framed in written language. Do you see that this is an issue that is more prevalent in authors that are non-English speakers? Because sometimes they're not too familiar with the way you make claim about significant or substantial.
Well, I would already pause, because I think that that's a huge issue, that you can hardly ever assume whether somebody is a native speaker or not, and that's actually a big problem in peer review. That, again, I see this on Twitter all the time, that people are like, just because my family name isn't European, or doesn't mean I'm not a native English speaker. So I would suggest extreme caution to any editor or reviewer to start assuming about somebody, whether somebody's a native speaker or not. And I don't feel like I could give a good answer to this question, because I don't have any data about that when I look at a paper.
Yeah. And I think there are plenty of people who grew up and were well educated in the United States who still make mistakes in their writing and the way they phrase things. So I don't, just following what Larry said, I wouldn't.
Right. So
to
answer this question from the audience, the editors, I just rephrasing, the editors say that there's no way to tell whether this is more prevalent in non-English speakers, because you don't know. Just because their name appear non-English, it doesn't mean that they don't speak English like a native. And also as Toby said, that even native English speakers can also be confused about these terms if they don't get trained properly.
Yeah.
I mean, the difference between significant and substantial is a important one that I wish that everyone would follow, even in more casual scientific speech, you know, like giving talks and such. Yeah, I wish that people would keep that difference in mind all the time in science.
Yeah. But yeah,
Right, so
I don't think that has to do with where you grew up and learned to speak.
I see. Yeah. So and then other aspect of research integrity, I think Toby mentioned this at the very beginning, is that how, if you can make the assumption that the data are solid, how do we flag that? Because sometimes when they submit, I don't think you have time to go through every numbers or go through their logbooks. So you have to assume that what they submit to you are honest.
Yeah.
So is that the way you deal with manuscripts and reviewing?
Yeah.
Yeah. So I remember there was one rather big scandal back in 2008 about acetic acid signaling, and it took several years from the paper was published in Nature, and it took several years for several groups around the world to not be able to replicate it, for the paper to be retracted. So is there any way or any rule of thumbs in the reviewing process that you can flag this before it's going to publication? Larry?
Well, there's been more of a focus recently on evaluating the figures, especially if there's things like gels where you can see actual image manipulation. You know, people like do things like you know, copy and paste bands, maybe like mirror them and, you know, try to blur them on the edges. So there's a researcher, Elizabeth Bick, that's very famous for this kind of image for forensics, but she has the uncanny ability just to see this like very clearly, where other people maybe have to be trained for it. But I think that in some polls that I've seen, some reviewers like barely check really even. So I think that we definitely need to be looking for at least this type of image manipulation that might be readily viewable. And then from a technical standpoint, there's more that can be done to look at the statistics. And Elizabeth has also pointed out cases like where if you look at the means and the absolute standard deviations given, that, for example, the like the coefficient of variation will always be exactly the same. So it's as if people, you know, are falsifying the data. So things like that you can see if you have like the data tables and things, and we should be checking that more too. But I think that most of us basically have the honor policy. We want to believe that people are being honest. And you know, that I guess that can be a fault in the process if you know, I can't verify every single step and what people did, even if they, I mean, if you find this type of data manipulation, then that really it's just an arms race, where then the data manipulator can just be smarter of how they manipulate data. One of the first things that I learned in the statistical programming language, was essentially how to create fake data using like the standard deviation and pulling out values from like a normal distribution, not to train you for how to falsify data, but rather to train you on the statistics. So the more that we have this type of race, yeah, I really worry that it just allows people who want to fabricate and cheat to get ahead, and this whole environment of having to hype your work for promotion, you know, and everything, it does lead people to look for the easy way out. There's also the recent example with the spider behavior papers, where it was just impossible for them to do that much work in some cases. So I guess when we evaluate the papers, we do need to think about, you know, whether it's even possible to do this work, if there's figures about gels and things, to actually carefully study it, because some of these manipulations can jump out. And if there are any tables of the statistics, to try to look for things like, hey, this data is just too clean and, you know, too equal.
Yeah. But I also think putting the burden solely on the reviewers I think it's not even going to work. And partly it's not fair to the reviewers. The reviewers are usually volunteering their time. They're usually on a deadline, and they usually have a few other things to do.
Mhm.
So yeah, a good alert reviewer may catch something, which is great. But I think there's a lot of institutional stuff that can be done, can and probably should be done, before the thing even gets submitted for review. I mean, for instance, my institution has the option of sending all figures for forensic screening before the paper is submitted. The institution covers the cost of that. So that, that we can all send in the figures to make sure that we haven't accidentally duplicated something or flipped something, or, something that looks like there's a gel splice. And they're often, I mean, there usually isn't, but I'd rather know about it myself rather than wait for it to turn up in some public forum. And that's provided by the institution as a service to the authors. And we could, there are some institutions that actually require that. We've taken a sort of a softer approach on it, but it could be required. So I think there are those kinds of institutional things that can be done rather than saying it's all on the reviewers to catch, if the third panel in figure 3 is a duplicate of the 15th panel in figure 10. I'm not going to catch that, even if I'm looking for it.
Yeah, here as well, we have a technical review where before submission, we should have another person who's not an author review the paper. So that could also help.
Yeah. Talking about the reviewing process, there's one question from the Q&A related to that. That is, sometimes we cannot just rely on peer reviewing process as the last line of defense. For example, for statistical quality control, because some reviewers don't even know what type of statistics to use. So should we have a system in place to check that, similar to your technical editor, Larry, to check the images and everything? So should the editorial boards or the journals have an in-house statisticians? Someone like Elizabeth Big, but not for imaging, but for statistics, to catch these things before going forward to the reviewing process. You see that's something that the journals would have to consider?
I think it's something that I don't know if only journals should consider, but as a community, for sure, we should consider that and what would really be needed. Like I said, I mean, I share data in our code, so if there's a way to just upload those and have it checked, for, I mean, with a lot of the quality checks, you can just kind of check data. For one thing, you can kind of just check the raw data statistically just to see if it seems plausible, like if that, like I said, if like standard deviations are always too clean and stuff, it just kind of points out. There's also ways to check for p hacking by checking to see if there's like too many significant tests, and I'm sure that all of our journals would find that if you look at the distribution of p values, they would of course be biased because we're not incentivized to publish null results and such. So I do think, I think that at least investing in things that can check the raw data, like if you could upload raw data with like the groupings, like, if it's different genotypes or different treatments, and then the software can automatically kind of point out if the data looks real or not, could be useful. But I think that ultimately it's just very difficult because the statistics involved can be very challenging for a lot of people, and there's probably no way to fully automate it, and the personnel resources are too high in a lot of cases to have a dedicated person. So it's a challenging problem that should be discussed more.
I think it's, there's certain sorts of data, RNA-seq data, for instance, where the analysis standards are they're pretty standard. They're
Mm.
in terms of the kinds of analyses you do, the kinds of ways you check things. And I could imagine a fairly automated system that would just go through something like that. But if you have say a field experiment with a bunch of different treatments and a bunch of different conditions and a bunch of different kinds of genotypes, analyzing those data can be complex, and it's going to depend in part on what the experimental design was and what it was designed to test. And it would be a big investment of time of a very highly skilled person. And then the cost of doing that is
yeah.
gonna be appreciable. And I don't want to get into the cost of publication, but it's not irrelevant.
No, yes, that's true. Right, I think I have one last question from the audience about the double-blind reviewing process. And the question is, should that be a standard, or are there any downsides? And maybe this has to do with, if you know the authors, you can go either too easy or too harsh on them when it comes to data interpretation. What is your thought on this, Toby? We're starting with you.
I think it's it's a very appealing idea until you start thinking about how to implement it. In that, I think most of us actually work in relatively small fields, and so that you're looking for a reviewer that knows the system, knows the questions, is sort of familiar with the big picture of that small field. And so they're also going to know most of the people who are working in the field.
Yes, that's true.
And so I think, for instance, any paper coming out of my lab, my guess is people would be able to figure out that it was probably coming from my lab, even if my name weren't on it, just because of the kind of work we do and the kinds of questions we ask, or they, narrow it down to me and a handful of other people. So I'm not sure it would really accomplish what it is designed to accomplish. And I've heard anecdotally that it's actually pretty hard to do. It's just to clean all references to the author out of a manuscript. But I don't know, Larry, do you have thoughts on that?
Yeah, it's something I know people have brought up and discussed. In some cases, it can get rid of bias. I think I saw a recent one using people's family names, just switching it, from European based to non and stuff can have big effects, even from
Sure.
names that are associated with women to men and stuff can have a big effect. So you can see the draw for it. But I also agree that it seems like those are all experimental cases, but in real life, I think you're right that people would typically know. You'd be able to figure out which lab it's from, and if there's a preprint, there's no the preprints are not blinded, so that's also been brought up, is that it'd be hard to control for that in a movement where we're basically pushing towards the publish then review type of mechanism. But certainly we should explore ways to decrease the bias. And I saw one of the participants might have mentioned in the questions that they feel like the reviewers on one of their papers might have been biased about their language purely based on their family name. So again, I just want to highlight again that that's one of the biggest problems I think is easy to fix, that editors and reviewers should be extremely cautious about making assumptions about where people are from and their language and things like that.
All right, thank you very much. I think we reached the end of our discussion today. Are there anything else that we didn't mention you'd like to add? If not, then I think thank you very much, Toby and Larry. I think it's very clear to us how harmful research misconduct and data over-interpretation are. And so I think the take-home message is for authors and readers alike is to stick to the data and have a healthy suspicion to data and interpretation. So once again, I'd like to thank Professor Elizabeth Toby Kellogg and Dr. Larry York today for their insights. And thank you, ASPB, for this very insightful, helpful event. Thank you. And back to you, Chris.
Thanks.
Thank you, Don. Thank you, everyone. Just a couple closing things I'd like to mention. If you want to see more webinars on Planté about journal content, we have several on the site. And coming up in 2023, there'll be some networking webinars planned by ASPB Planté Fellows. And you can follow at Planté_org on Twitter for webinar and event announcements. And don't forget that access to ASPB journals is free with ASPB membership. So if you aren't a member, we encourage you to do so to enjoy those benefits. And I want to thank you again, all of you, for joining us for this special event. And thanks to Don, Toby, and Larry for making this webinar